So this next session that we'll be talking about, that's going to be about is on something called antimicrobial resistance. So drug-resistant infections have been becoming a major public health concern all over the world. And microbes are increasingly becoming resistant to the antimicrobials that were supposed to target or arrest their growth. For example, infection-causing bacteria are now becoming resistant to the very antibiotics that were supposed to kill them or inhibit their growth. And this is something that has arisen largely because of a rampant overuse of antibiotics. And in this concerning situation, scientists are trying to find out that is there any alternative that exists for these antibiotics. And an unlikely ally has now appeared in the form of a good virus, a so-called good virus called a bacteriophage that can target bacteria. So to explore the science, promise, and lived realities of these so-called good virus, today we have Urmi Bajpai, a microbiologist who has been taking a closer look at phages, and Pranav Johri, who has himself undergone phage therapy, in conversation with the moderator of this session today, Sara Iqbal, who happens to be a science communicator, an engagement strategist, and co-founder of Superheroes Against Superbugs, a very unique initiative that has been actively engaging with young people to educate them about what is this global threat or growing threat of AMR. So with that, I'd like to request Sara to please take this discussion forward.
Thank you so much, Anushila, for that kind introduction. And it's really nice to see you all here for this session that I hope is going to be engaging, interesting, informative. A very big thank you also to the Science and City team at the Hyderabad Literature Festival, because they have been creating this space to talk about AMR, which seems so abstract, technical, something that makes you wonder about why should I even be listening to these guys. But they've been consistently over the years creating this space, so I'm really thankful to them. Even though we'll be talking about antimicrobial resistance, but in a very different context, and the context has already been explained wonderfully by Anushila. Before we begin, I just wanted to get a sense of, what you think. I mean, are all viruses bad? Any show of hands, are all viruses bad? Anyone here? Okay, there's one person there. Hope to change your mind by the end of this session. Yeah, which is impressive. Maybe because there's a lot of science, medicine folks here, but that even if they're not, it's quite encouraging. So the title of today's session also has been very intentional. We are calling Good Viruses. And it comes from the wonderful popular science book by Tom Ireland called The Good Virus. It's about bacteriophages, which are the bacteria-eating viruses, as Anushila mentioned. But we've borrowed this title very intentionally because our work in science and health advocacy, especially when we talk about AMR and infectious diseases, we tend to focus on the dangerous microbes, the ones that are causing these stubborn infections that are getting harder to treat. And without really meaning to, we end up telling a very lopsided story, which is that all microbes are villains, where every bacterium is suspicious, every virus is a troublemaker, every fungus is a bad omen, but and us, we humans, are constantly at war with the microbial world, trying to either exterminate, control, or dominate these microbes. And the recent COVID-19 pandemic has intensified this instinct and this emotion as well. Even in literature and pop culture, we always see microbes as the bad characters. But the microbial world, I think those of us who have studied it or know it, we know that it's not a battlefield. It's a diversely vibrant ecosystem that we are all intricately connected with and that we are part of, that we also influence. We have as many microbes in our body as we have human cells, which goes to show how important they are. Interestingly enough, the genes that help the human fetus to connect to the mother's womb, the placenta, is also made up of viral genes, or genes that have been borrowed from viruses. So we're really a product of viruses and microbes and bacteria. Which should really make us think, rethink our relationship with them. But having said that, of course, there are microbes that harm us. But there are also many that protect us, sustain us, live in us, evolve with us. And in the case of the good viruses that we'll be talking today, bacteriophages, some even come to our rescue. And I think this becomes very important, I think, in the context of AMR that Anushila also mentioned at the start, that if antibiotics stop working, then what do we have? What other alternatives do we have? And this is where these bacteria-eating viruses, which scientists call bacteriophages, become very important. And as the proverb goes, the enemy of my enemy is my friend. So in this situation, the bacteriophages have become our friends, or at least we're trying to explore that. Another very interesting fact about bacteriophages before we dive into the conversation is how many of you have heard of the magical or the healing power of Ganga, the river Ganga? You must have, or of some water body, right? Or the Ganga Jal. So the interesting story is that back in the 19th century when they noticed, they observed that a lot of the infectious diseases, cholera included, would stay upstream of the river and would not be seen downstream of the river. So obviously people used to wonder what would happen, how is the infectious disease not flowing? But at that time, there was also this understanding that infections don't pass through water, but they pass through putrid air, like impure air. So that was also a widespread belief. But in 1892, an English scientist, Ernest Hanbury Hankin, traveled to India to study cholera and other infectious diseases. And he noticed that when you boil the water from Ganges, when you don't boil the water from Ganges, it kills the cholera germs. And when you boil it, it doesn't. So this was probably the first observation of the bacteria killing, properties of bacteriophages. It was published, but it was only in 1915 or 16 that bacteriophages were discovered by scientists. And since then, I think Urmi will tell us more about where we've come this far from there. So we can see that through phages, we want to actually explore how we think of viruses, how we think of nature, what options do we have today in the age of drug-resistant infections? And to help us make sense of all of this, we have two excellent people with me. Urmi, who explores the science of phages. Pranav, who's lived through AMR and has taken phage therapy and now helps others navigate this deadly disease and also helps them understand phage therapy and access phage therapy. They're probably also the reason why I know whatever little I know about phages or I have interest in phages, so thank you for being here. Just one disclaimer is that this is not going to be a session where we will dole out any medical advice. We are just going to explore this fascinating world of bacteriophages, these viruses, and also the seriousness of AMR. Why should we be thinking about it? Why should we be doing something about it? So with that, Urmi, perhaps we can start with you, and I should admit that when I first met you and you told me that about your interest in phages, or I learned about your interest in phages, I was like, why would she be, of all the things in the world, why is she looking at phages? Of course, I know that better today. So maybe you could tell us a bit about your interest in phages, what really drew you to them, and also how do they work? What makes them so special? What gives them the good virus tag?
Thanks, Sarah, for this lovely introduction and setting the stage so well, and I thank all HLF organizers for this opportunity. Phages have been long neglected, long forgotten, so wonderful to see this kind of excitement and attendance and taking up this session. How I got interested, actually, the way Sarah asked that why is Urmi interested, even I was not interested early. I was doing, my work was on finding new drug candidates for TB. And I happened to be in the US for something else, and I had some time and I attended a workshop. And there where I found that when I learned about phages hands-on, like we were doing as a life science students also, but it did not somehow caught my fancy. And then around the same time, because my research was on drug discovery in the background of AMR, and suddenly I thought, it was being thought earlier also, that if phages have such killing properties against bacteria, why aren't they being used for treating them, in place of antibiotics or as an adjunct to antibiotics. So that's how I got interested, because my work was already on AMR, but we were not looking at phages. Now, why phages are suddenly, like in our part and in the West, they have got attention, but in East Europe, they have been working as antibiotics for almost like now more than a century. So it's somehow I want to tell this, it looks like that phages are new. They aren't as new, they have been there since forever. They were kind of observed, as she mentioned, by Hankin in the late 19th century. And so, when they were discovered before antibiotics, and so in 1920s and 30s, phages were being used for treating bacterial infections. And then, antibiotics came in. Now, antibiotics are very simple drugs, as you know. They are very chemical entity, very defined. You can characterize them well. And suddenly, physicians and scientists, they preferred static antibiotics over living phages. And I say living because they have their own mind. So in general, people avoid people who have a mind of their own. So they thought, let's recruit antibiotics as killers of bacteria. And now that we see so much of resistance has occurred, and we are actually running short of antibiotics, and there are no new drugs in pipelines, or very few. So then now we are returning to phages and understanding their potential. And the ability of them to kill phages, different from, if I may say, different from antibiotics, is that they are not as, like if I call antibiotics as scattergun or grenade, who have broad spectrum ability, phages are very precise, very specific to their target. So we haven't we do look at antibiotics as wonder drugs, and of course they have made our modern medicine very improved and impressive for public health, but the collateral damage which antibiotics do to us, to our health, to our microbiota, that was a bit ignored because, they were so wonderful otherwise. And now that phages are so specific to their target bacteria, and they're so abundant that, there is no kind of, we can say that there's an inexhaustible supply of phages in nature. So we are not going to run short of them for sure. We just need to pick them, study them, and use them as therapeutics. So that's where I feel that they offer so much potential, but the major limiting factor I find, or the bottleneck I find, is extremely limited awareness and skepticism. And that's why perhaps they haven't got the limelight they deserve. But I'm glad with such fora, I think they will pick up. Thank you.
Thank you, Urmi. So how are they different from, say, COVID, the coronavirus, or influenza virus that causes cold? So what are the differences? Why are those viruses bad and this one is good?
So corona is very fresh in our memory. So coronavirus is something which infects humans. So, it causes disease to humans by infection. Whereas bacteriophages, as the name suggests, these are viruses which infect bacteria, not humans. And therefore, we are safe. So they are generally regarded as safe because they don't cause any damaging effect on humans. And so now you have something which has very targeted killing spectrum of only killing bacteria which cause us disease, but not to humans. And that's what makes something which is causing me disease, and a virus comes and kills it. Is it not a good virus? And that is why we say that it's a good virus in the context of therapy, that they help us get rid of infections by bacteria, very specific to bacteria. Also, they have several ecological roles, but I don't know if we need to discuss that. For example, bacteria, because of their ability to kill phages, so, we'll think of that, why are they there in nature at all? And it is because, they tend to keep a check on bacterial population. You can't let them grow because bacteria are very prolific dividers themselves. So phages' primary job is to keep bacterial population in check. And also by killing them periodically, they also keep the biogeochemical cycles going on. They release nutrients and they make it available for organic growth. So in a way, they kind of, modulate the community, microbial community, which is very important. Otherwise, the ecosystem will be very imbalanced. So they have a very silent and invisible role in maintaining these ecosystems, which we are so used to of.
And we have bacteriophages in our bodies too?
Yeah. And also because phages are like viruses. So as I said, as Sarah mentioned, and most of the audience did not think of any good virus. So typically we associate virus with disease, whether for plants, like, the first virus which was structure was solved was tobacco mosaic virus in plants. And in humans, we have so many viruses, latest corona, everybody knows. So we have, as she mentioned, that we have as many human cells as we have microbes in our body. And those microbes include a virome, which is like viruses. And in amongst those viruses, the most predominant are phages. So they are in our system. They are, in fact, I would say, kind of gatekeepers. They do not allow opportunistic infections of opportunistic bacteria to invade our system. So they kind of act as gatekeepers. So silently, they are anyway contributing a lot to our health. And, but now that because of their antibacterial properties, we can recruit them as, I would say, living antibiotics.
Very interesting. And do they have any other application, or there's any other interesting science that maybe your lab or other labs in the country are looking at of phages?
Yes. So if I look at bacteria, they are like parasites. They predate on bacteria, and so I'm saying they kill. So how do they kill? So what they do is, I mean, initially when I was like a student, I used to find them very mean. That they are so mean that they, they just go latch onto an unassuming bacteria. Bacteria is sitting quietly somewhere. They latch onto it, just gets in, they slip in their genetic material, and then they hijack the whole machinery, direct the host to make copies of themselves, and they're going to then kill them and get out and kill more of them. So looks like the crazy and very selfish, killers. But now we realize that this is important to keep the bacterial population in check, to keep the ecosystem as it should be, and also not letting any bacteria grow in abundance. So you have to keep their numbers in check, and that is why it is. I forgot, what did you ask actually? What science? So to kill the bacteria, they make some enzymes, at the end, when the phages have made progenies of themselves inside a bacterial cell, they need to get out of the bacterial cell to infect more, right? For that, they need to do some cuts in the cell wall of bacteria, and so that they can get out and infect more. And what are the enzymes they make to get out? Those are called as lysins, as the name suggests, proteins which lyse bacteria. And so these lysins are now, it's a relatively new science and technology. It's started in 2000, so about 25 years ago. So my lab is working on finding bacteriophages against mycobacteria, which is the tough bacteria to kill, and also finding enzymes which these phages make to lyse cells. So we call them enzybiotics, means enzymes as antibiotics, or protein antibiotics. So these are the areas which, and also if the students of science would appreciate that bacteriophages have immensely contributed to molecular biology, we know. We couldn't have done genetic engineering if we hadn't known that bacteria make restriction enzymes to ward off phage infection. So the CRISPR-Cas, restriction enzymes, these all are very good tools today in editing, genome editing, and also in genetic engineering. Also that DNA is a genetic material. This came from research from phages, that it is DNA, not protein, which is so very seminal contribution of phages to science has happened, but they were used as tools. Now, given their lifestyle, can we use them as antibacterials is the question.
That's really fascinating. Thank you so much, Urmi, for that. So Pranav, if I can come to you. Pranav is actually a publisher. He's in the business of books. But he's not at the Literature Festival to talk about books. He's here to talk about AMR and bacteriophages. Because unlike Urmi, you encountered phage not through research, but through a personal experience. It was not some abstract technical concept for you, but something really alive, and something actually that you probably saved you. So could you tell us a bit about your journey with drug-resistant infections? How did you come across phage therapy? How did that work out for you? Just help us maybe make phages a little more real in the human world. How did that work?
Thanks, Sarah. I'm actually sitting here because of bacteriophages. 10 years ago, I had a multi-drug resistant infection, and I wouldn't be sitting here right now in front of you if it weren't for phage therapy. I went through multiple courses of antibiotics, oral, IV, then combination treatments of oral plus IV, exploring synergistic impacts of different antibiotics, and then ultimately, at the end of the last course, which my doctor had called, we're going to nuke your infection, which was amikacin IV with ciprofloxacin oral. At the end of it, he was like, we've tried out whatever antibiotics we could to deal with your infection. We've explored combinations. We've tried everything that we could. And unfortunately, the infection seems to be non-responsive. So we've kind of reached the end of the road in terms of being able to treat the infection. We're going to just have to figure out a way to manage it. So I was like, how do you manage an infection? I mean, by then I had been living with a fever for 6 months. I'd become really weak. I'd had side effects in terms of the pains, and the morbidity from the infection was really impacting my quality of life. I couldn't work. I couldn't sit. I wasn't able to sit like this. I mean, if this was 10 years ago, I would be wearing 2 sweatshirts on me right now because I would just feel chills all the time. So reaching that end of the road, and the doctor was like, yeah, we'll just have to manage your condition. And he was like, you'll just have to find a way to live with it. So that's the position I found myself in. And I was like, okay, it was a really difficult kind of sentence to take from the doctor. He was a HOD at one of the top hospitals in Delhi. And then went through a really difficult time for a few days, kind of dealing with, the typical questions that one deals with, like, why me? Why is this happening? What is going on? But then I was like, you know what, antibiotics, we all studied in school. Alexander Fleming discovered it at that time it was about 70 odd years ago. I was like, how did people deal with infections before that? There must be some other way to deal with the antibiotic infection, with bacterial infections. And that got me started down the path of researching alternative ways of treating bacterial infections. And there was a paper that I was reading on PubMed, a research paper, that was tracing antimicrobial therapies. And that spoke of phage therapy as something which started coming online, coming into the mainstream gradually in the 1920s and 30s. Came across, Eli Lilly had come up with some phage products at that time in the 20s. Or was it 30s, against typical infections of that time. So they'd come up with something against salmonella. They'd come up with something against syphilis, phage products. And then, of course, once antibiotics got discovered, and then soon after, this was in the middle of World War II, and then the world got split between the Iron Curtain and the West and the East, and everything that was happening behind the Iron Curtain remained behind the Iron Curtain. And it just so happened that I think a lot of the phage research that was happening was all in the Soviet countries. And then the West, and thus the rest of the world, all adopted antibiotics. And so that paper really opened my eyes, and I started looking into phage therapy as a potential path for me, and realized that there were mainly 2 institutes at that time in the world which were actively involved in therapy. One was in Georgia in Eastern Europe, one was in Poland. Reached out to both. The one in Georgia seemed to be the most experienced, had the largest phage library, which is their repository of phages that they can use to treat bacterial infections, and reached out to them, and finally 3 months later I landed up there for my treatment. And they were like, 1.3 billion people, you're the first person from India who's approached us for phage therapy. So yeah, I was the first person from India, and that's how I discovered phage therapy for myself.
Thank you for sharing that. That's really interesting. I mean, for you to be the first recipient. So what does phage therapy currently look like in India and globally? I mean, could you give us a snapshot of that? I mean, where are we headed with that?
Okay, so in terms of India, we've had a long history of phage research, and there's been research happening for 25 odd years. But in terms of applied, and that's the case with a lot of countries, where phage research has been going on. Phages are brought up to a certain level, but I think the lab to bed transition has not happened. The lab to bedside transition has not happened. It's remained in the scientific domain in the labs. But in the last 10 years of, especially I think post-COVID, there's been an acceleration in terms of different countries, different institutions looking at application of the therapy. So even if I chart our own journey with phage therapy, so I had my infection 10 years ago in 2016. once I was able to overcome it with phage therapy and once I recovered completely from the infection, we established an initiative called Vitalis Phage Therapy with the aim of making phage therapy accessible in India and increasing awareness of AMR and phage therapy as a way to combat AMR in India. And initially, I would say for the first 4-5 years, mostly people who were contacting us were patients who had gone through multiple antibiotic courses, failed treatments, and then the medical kind of system had kind of given up on them. They had been told that, you'll just live with the infection, or they had been told that amputate karna padega if it was a wound infection or something like that, or they had been told that in some cases, where the infection was not so apparent, they'd even been told, oh, it's all in your head, like go refer to a mental health professional and stuff like that. So where the infection was not apparent, it was usually it used to be individuals, patients who had been kind of given up on being able to treat their infection with the medical community. In the last 4 odd years, we mostly get now contacts from hospitals, from infectious disease specialists for their patients. So the awareness of phage therapy has really gone up. And I mean, in terms of application, beyond human application as well, agricultural application has started happening. There is application in terms of animal, livestock. Yeah, so there's a greater awareness, and certain countries, I would say, have really leapfrogged, going from research which was not even at our level, say 10 years ago, to now having very well established phage therapy consortiums where they've brought in the research institutions, the medical institutions, the doctors, all of the stakeholders, science communicators, to create awareness. And those consortiums have really started increasing application of the treatment, awareness of the treatment.
Great, thank you. That was really helpful. So, Urmi, what do you think is the barrier for making phage therapy or research in phages a lot more, to strengthen it in India? What are some hesitations on a policy level or maybe on the research community level? What is your understanding of that at this point?
Yeah, I agree that there is a hesitation, and the reason is that, as Pranav mentioned, that in the last 10 years there has been awareness made in this direction. Now, there's still a lot we don't know about phages and phage biology. So research has to go on because that will supplement, their candidature as therapeutics. So the gaps that we don't know, the primary concerns which I feel people have, one is what will be immune response. And second is how safe they are. And third is that especially for gram-negatives, that if phages lyse bacteria in large numbers, so typically, these gram-negative bacteria, they release toxins, and those endotoxins can cause septic shock. So these are the concerns which in general people have. But these are not like, for example, even antibiotics can cause mass lysis and can cause release of endotoxins. So it's not, and you can always, modulate the dosage of phages to prevent this from happening. It's not really a end of all thing. And second, about immune response. So far, there are a lot of compassionate studies done all around. Like Ilyava has been practicing phage therapy for 100 years.
Ilyava
is a Georgia in Tbilisi. So the one which he mentioned. So in Tbilisi, it was erstwhile USSR. So they have this institute, and they have been practicing phage therapy since about 100 years, and now medical tourism has, I think, quadrupled because of AMR. And so we find that actually there is a consensus that phages are generally safe. There are no reports on adverse effect. At the most, sometimes, there's a variable treatment, which means the variable response to phage therapy. That is more to do with the people than to do with phages. And also, they do bring out drastically the bacterial numbers. It is also reported, like there's one scientist in Belgium. So their group, as he was mentioning, they have a big group, and they have 100 cases they recently reported in a retrospective study. They were doing phage therapy for many patients, and then they found that what were the pluses. So they found 77% cases showed a lot of improvement, which is a very promising number. But the thing is that putting together the research part first, acceptance of phages as credible alternative or adjunct to antibiotics. Then some dialogues going on in the circles who matter, for example, those who write policies like CDSCO and DGCI, etc. And third is bringing together the people who are working on phage of different aspects, and then making a roadmap. What will it take? So, the roadmap is absolutely doable. I presented many of my talks that what is the roadmap to make phages, develop in India. But right now, the absence of dialogues with the people who matter, because, phage people, they have phages. Most of the people I know, including myself, we have our own phage repositories. We have been isolating for years now. But can we use them as therapeutics? That is the major refrain. And that is because, you don't want to scare yourself or to anybody that if my phage, causes some damage to somebody and then, phage therapy goes back by decades, like it happened in gene therapy. Once a gene therapy, they had used adenovirus as a vector. And when they gave adenoviruses, it does not give you long-term immunity. So when an adenovirus-based gene therapy was administered to a young patient, and on the second or third dose, he had a severe immune response, and that patient unfortunately died. And that pushback gene therapy, people just abandoned it because they thought it's so bad. So that's why people are very cautious. And I can assure now with our understanding and the examples, phages are far, far safe than anything else. In fact, the kind of collateral damage antibiotics do, phages are so safe. It is just that we need to put all the information together to capsulate them as therapeutics. That is what is, I think, holding us back. Dialogues.
That's interesting. Yeah, that's very interesting. Also, if bacteria has become resistant or becomes resistant to antibiotics, wouldn't it also become resistant to phages?
That's a very good question.
And Pranav, were you not worried about that? How did you explore that aspect also in your, so both of you could respond.
So yeah, actually, I had a multi-pathogenic infection, and one of the bacteria was resistant to all of the standard phage preparations or cocktails, as they're called. But as Urmi mentioned, phages are infinite in nature. So when you hit a situation like that, with say an antibiotic, you've hit the end of the road. But with phages, you can actually go out, isolate phages against that particular strain, come back and prepare it into a therapeutic. So you prepare it into a cocktail. And that's what was done in my case. One of the strains, they had to actually go and isolate phages and make a customized phage cocktail for me. And I think just to add to what Urmi was saying right now, I think our system of medicine is right now in a transition where we are going from standardized one approach for the whole population to more personalized medicine, and phage therapy fits more in the personalized medicine place. So I think right now we are in a place where our frameworks, whether it's regulatory or drug control, are all for standardized medicines, versus whether we look at one size fits all. And in terms of the future, we're going towards personalized medicine. I was having a conversation yesterday with a urologist and his patient who has a UTI, got it from a catheterization in the hospital. And they had the same questions that, what impact would this have? And at the same time, the patient was complaining, the antibiotics have messed up my gut and I'm on diarrhea. I have diarrhea all the time, and I'm going through weight loss. I've lost 6 kilos, blah, blah, blah. So the doctor was like, we are really worried about the impact of phages. And I was like, the phages against the strain was Pseudomonas aeruginosa. I was like, the phages against this particular strain are already in his body. If we were, say, 20 years from now, we would possibly take his urine sample, isolate phages from it, make it into a medicine and give it back to him.
You can do now also.
Right, I mean, if as the frameworks catch up. I was like, the phages are already there. So antibiotics, you're acknowledging the impact that they're having on him and his other aspects of his health. The phages are already there. So we as Urmi said, the science is already, and we mentioned about numbers. The Belgium, they've just done 100+ case study they've put out, but they've treated now 238 patients in that lab MCT. Since we started in 2018, we've had more than 330 patients who've been treated with phage therapy. I know similar numbers from the US, Taylor Labs. They've also 100 plus, I think, patients. So there, yeah, so there's enough number of like cases now where it cannot be dismissed as anecdotal or, oh, this is just a one experiential thing. There is enough evidence for it to move from just being anecdotal evidence to actually moving over to.
And the numbers are low because there needs to be a policy shift, there needs to be a mindset shift on the medical and the political.
Yeah, and even the awareness, as Urmi mentioned. I
And,
mean,
yeah.
right now we have had people, there was this particular case where an elderly gentleman had a catheter-associated bladder and kidney infection, and he had already been put on colistin. And then his family approached for phage therapy, and the infection actually started coming down, but the colistin had damaged the kidneys to a point where he died of kidney failure. So his family was like, we wish we had learned of phage therapy 3 months earlier, 2 months earlier, before medication which would have damaged his kidneys could have been done. So it's not something where we're like, no one's making the point that phage therapy is going to replace antibiotics tomorrow, but it definitely has a role to play as a supplementary form of medication for controlling infections, for dealing with infections, especially where antibiotics don't work.
Yeah, I want to add the biology part here because, I call them Tom and Jerry because, phages and bacteria co-evolving. Both need each other. Nobody will let anybody get past each other. So if even in real time during therapy, if resistance occurs, and it does occur, so then, because they are co-evolving, phages find a way to, infect in some way. And as I mentioned and Pranav mentioned, that there are so much, the numbers are so high that we can always recruit new phages against the same strain. And one more interesting thing and very promising thing about using phages with antibiotics is that sometimes when bacteria gains resistance to antibiotics, phages come in. And if bacteria gain resistance to phages, then, there is a trade-off. And now the resistance to phages comes at a fitness cost to bacteria. And so bacteria is in catch-22. What to do, If I change my receptor, my phage can't bind. But because I've changed my receptor, now my bacteria can throw antibiotics and hence cannot gain resistance. So, when we use them together, in fact, recently, the same team, they are working on Ukraine, and they are developing phages for patients, the soldiers infected in the war and those infected with those drug-resistant. So they found that bacteria lose plasmids. The plasmids also are extra chromosomal elements which impart resistance to antibiotics in a big way. And what they found is when the phages are infecting these strains, and over a course of time, bacteria develop resistance to phages, but they also lose plasmid. Plasmids get cured, and therefore suddenly the bacteria becomes susceptible to antibiotics.
Plasmids, I think, might be lost on a lot of people.
Plasmids are like the extra like not the regular genetic material. They are like accessory genetic material, which give them some additional advantage. And typically, bacteria have those extra genetic material to withstand antibiotic pressure. So they can resist antibiotics because of these. So you imagine that if phages are there, they die of infection. If bacteria develop resistance to phages, then they die of antibiotics. So this is what needs to be understood in terms of biology, that how the trade-offs work and how the fitness cost can be considered to make sure that one of the two will work and, we'll be able to. Exactly.
That's fascinating.
Yeah.
That's really interesting. So then, like Pranav also mentioned in one of the examples that one of the patients had diarrhea because the antibiotics is well known that they target both good and bad bacteria. So how do phages learn to distinguish, or how do phages not kill the good bacteria? This is, by the way, a question from my mother. When I asked her in the morning, what should I ask you guys? She was like, how do phages know which is a good and which is a bad bacteria to kill?
Yeah,
So
absolutely. Yeah, so phages, they latch onto some receptors, like antenna we have, like earlier, when we used to fix TV by fixing antenna, remember? So likewise, bacteria have those antenna-like things on their surface. And phages when we administer phages, we will administer phages only for those bacteria which we want to eliminate. And therefore, our phages are only picked up for those. And those phages are so specific to one type of bacteria that in our gut or anywhere else in our body, there are different types of bacteria which are important for our health, they are called commensals or good bacteria. And phages will not touch them because we haven't pick those phages which can target those other bacteria. Our focus is on the disease-causing bacteria. So the phages which we administer are only those which are for this bacteria, and other bacteria don't have a receptor to bind to phages. And therefore, it's like precision, like, snipers. They just do very targeted killing. Like, you throw a grenade and it kills everybody, a lot of troops. But if you throw a sniper, it will just kill that bacteria. So we again use the biology, understanding of biology, that which phages are specific to this disease-causing bacteria. Just grow them, develop them as therapeutics, and deliver them. And this specificity makes them very unique, much more advantageous over bacteria antibiotics in long term, because we have been taking antibiotics. We have been, of course, getting fined. I'm extremely grateful to antibiotics, no doubt about it. In fact, major part of my research is discovering new antibiotics. But at the same time, the collateral damage they do to us, because of their ability to kill more bacteria than they should. The damage, especially in neonatals, in this young children who are administered antibiotics, their cognitive abilities are impaired, they develop asthma, their immune system is compromised. All of this shows up later in life. We don't even know. At that time, child gets fine. So this specificity helps phage go and kill exactly the target without affecting anything else which is good for you, which is the best part about phages.
I will check if you got that later.
I really hope you got this. I really hope you got this.
Okay, so another question. I wanted to do a quick rapid fire, but I've just got a time check, so we will maybe do it slightly differently. So the first question is, are viruses living or non-living?
I would say
Most basic, right?
Yeah, so I would say that since they have their mind of their own, so like sometimes even animals go on hibernation, so they just freeze, and then when they thaw, they start functioning. Likewise, when Stanley had first crystallized virus, he could see that these are like sitting crystals, absolutely inanimate, fixed, static, and when you thaw it, they become liquid and they are infectious. So it's like, actually they're called as something which have both the properties. They have properties for destruction, they have property for maintenance. And so therefore I would say, call them towards living. They need assistance, we all need assistance. All of us we live by some or other assistance. So what if they need?
All right, we'll take that from you. Pranav, if, I don't know if you're already thinking of writing a book, but in case you are, what would be its title?
So in publishing, we have a concept of working title. So working title would be, I'm writing this book because I'm alive because of phage therapy.
Right.
But yeah, I mean, actually I thought of this when I was coming out from my infection with phage therapy. I was like, this is a really good virus, and we all associate viruses with just negativity. And I was like, wow, there are good viruses as well. And the good virus got taken as a title, and now it's also the title of the session.
So yeah, so we have, great, wonderful. Look forward to reading your book. So if there's, actually I want to touch upon AMR before we sort of move to the audience question. So, AMR is often something that's blamed on misuse, like we're overusing, misusing antibiotics, that's driving antimicrobial resistance or drug-resistant infections. We're using it in humans, we're using it in farming, agriculture. Pharmaceutical companies are dumping their, antibiotic-laden waste in the environment, and various things like that. Pharmacists are giving antibiotics without prescription. Doctors are giving antibiotics even if it's not a bacterial infection. So there are a lot of these reasons that are given for why AMR is becoming such a big problem. I just want to get your thoughts on whether you see that the prevention of AMR, is it up to people, or is it really also got to do with systems? The systems in place that would regulate or manage these actions or these behaviors. So just some very quick thoughts from you on how you see AMR as a problem. Is it only a problem of people, or is it also a problem of the systems we have?
I think it has got to, because people will do what system allows them to do. So the compliance from people, and in fact their ignorance also is a catalyst, because you want quick fixes. And also system, if it stringently kind of makes compliance or enforcement is better, then of course people will find a way around. I mean, if it's all about enforcement, but at the same time, we also have to see that, in typically, if you fall sick and you take antibiotic without prescription, you have a pressure to go join work. Do you have the luxury to stay home and, take 1 week off from work? So those social factors also have to be brought in while we are discussing this. What is the work culture? What are the compulsions of people to take antibiotics? But overall, I would put responsibility on the system for finding adherence, for stewardship, and gradually people will definitely follow it, I'm sure.
Absolutely. Thank you. Thank you for that.
So I think, as they say in general for India, like what you say for India, the opposite is also true. So sitting here in Hyderabad, we are talking about the problem of excess. When you're talking of antibiotics, excess, overuse, right? We go 70, 80 kilometers north, south, east, west from here, and suddenly the problem will be of access, where they won't have access to antibiotics. So in India, we have both the issues. We have the problem of excess in Delhi, Bombay, Hyderabad, Bangalore, and then you go 100 kilometers away and you'll have the problem of access, where they actually need antibiotics to save a person's life or save a limb or something, and they don't have access. So it's a very fine line which has to be tread because we have both the issues. Two, I think, there's so many aspects too, as Urmi said, like, ideally for a viral flu, why would you take antibiotics? But then a flu will take, say, 5 to 7 days, and you potentially could have a minor secondary infection, which your body would be able to overcome. But then can you take 5 days off from work to be able to allow your body to heal through that? So you can say, put the blame on pharmacists, that, oh, this pharmacist outside is openly dispensing antibiotics without prescriptions. But that guy is probably at the end of the day making, I don't know, like making $1,200 a day, and then he has to run his household, feed his family, send his kids to school. So there's economic compulsions as well there. So the whole, I think it's a issue which is way deeper than just putting, oh, iski galti hai, ya iski galti hai. It's a systemic thing, as Urmi said. I mean, we have patients as well where they'll say, oh, I went to this doctor, he or she didn't give me any medication. I went to another doctor, he or she gave me medication, so the second doctor is much better.
Much better, yeah.
I mean, the first doctor was actually saving your health by not, say, over-medicating or mismedicating you. But the doctor gets, points deducted for that. So it's a very, very systemic, cultural, attitudinal thing, and I think probably needs a very holistic approach to it. It's not a quick fix thing, and possibly it's something which, as we evolve as a society, probably it's something that will happen over time, but there's no quick fix for this, I think.
Thank you so much for sharing those really important thoughts. I wanted to highlight that.
When we talk of AMR, I mean, it may not be very technical, but prevention and hygiene, these two are so important. We can really curb infection. Then AMR will itself automatically be reduced. So, bringing those also into compliance or enforcement, I don't want to use, but, making people aware about hygiene can prevent so much that consumption of antibiotics can go down.
Yeah, that's great. Yeah, so prevention is what,
Just as the dialogue, I think it's gotten stuck in so much jargon, which is difficult to understand. I mean, AMR as well, antimicrobial resistance, okay, like, let's go out and ask what does that mean to anyone. Like, they'll be like, pata nahi. But like, if you ask, say, what will you do if antibiotics don't work? Antibiotic aapne li hui hai? Haan, li hui hai. What will you do if it doesn't work? Oh, suddenly the thing hits them. So I think the whole framing has to also become in a more relatable way.
That's an excellent point. Thank you so much. Yes, so it's a perfect segue into question and answer, and we already have one question there and another at the back. Oh gosh, whoa. All right, we'll see how many we can take. Kritika?
So the question is about the range of infections which are addressable by phage therapy. I'll just give a short answer and then I'll hand it over to Urmi. So any bacterial infection can be tackled with phage therapy. The quicker you approach it, so if it's say a UTI and you allow it to become a urosepsis case, then the later you approach it, the more challenging it is. But that's true for any treatment. The earlier you approach it, the higher the chances of being able to address it. But any bacterial infection can be treated with phage therapy.
Yeah. So in practice, primarily topical are the most topical, and pulmonary gastro-infections. These are the ones which are covered the most. And the urinary, or I would say urologist, I mean, all those things. And the ones which are not as much done is systemic, as you said, because there is not very good understanding of what kind of response, what kind of endotoxic shock it will create. So systemic infections are relatively few, but we have started seeing even one. There's Professor Gopalnath from BHU. He has recently treated a patient who had systemic infection with Acinetobacter. And those infections, which we call as where bacteria goes and sit inside a human cell. So we call those infections intracellular. Those are difficult to target. So these are the two which are relatively less, but now we see more news coming on those as well. But otherwise, the rest were in these categories, the gastro. And
Hello, yeah, this is Divakar with Redfire. We work on artificial intelligence. Whole healthcare is moving from addressing cure to the preventive now and predictive things. So how any research outcomes or anything are we working on these specific virus interactions and you're getting infections because so much of data is available. Preventive sort of viruses which will help you to more immune, at least we don't go to the cure kind of a scenario. Is any outcomes like that with you? Please share with us.
so for artificial intelligence, did you say yeah? So for artificial intelligence, primarily because, yes, you rightly said that so much database is there. So it is being used for predicting, like ma'am asked, that how do you choose a phage for an infection? So, based on the bacteria and phage databases now, because so much of genome sequences are now available, one can predict, there are algorithms which can predict which will be a best match for this infection. And second, for the prophylactic use, you're saying that if we pick phages which can prevent, as you're saying, so for that, phages are used prophylactically also, but at this time, the thrust is more on therapy because of the MR. Yeah.
Hi, good afternoon. Hi, good afternoon. You were talking about specificity of bacteria and phages. So I have learned about enzyme and substrate, which works as a lock and key model. So my question is, does bacteria and phages also work in a lock and key model? Like, is it very, like, for one bacteria, only one specific phages, or it can be
That's a good question, because most of the biological interactions are very specific, like you said, enzyme. But as you would know, that a lot of enzyme substrate interactions are, besides lock and key, they're also induced. So in case of phages also, there are receptors, but there might be, to begin with, fixed receptors. But as I said, that they are co-evolving, so receptors keep changing. And bacteria also start finding them. I mean, the bacteria will change the receptor, because bacteria has learned over a period of time that this phage is going to infect me via this. So they evolve and they change. So then phage comes like a Johri and says, okay, let me find something else I can latch onto. So it's a very dynamic interaction. I wouldn't say it's fixed. We have identified key receptors, which largely are used to latch onto, but we can't say if bacteria modify them, then bacteria cannot get infected by another. And so every bacteria will have several viruses against them. Yeah.
sniper part that you spoke about, then do you have a targeted kind of a, I mean, using technology, the AI-based technology, do you use a targeted approach? Like you mentioned in case of the sniper. Do you do that? That is one. And the next is about preventing the virus, like what you were explaining just now. Do you use technology to prevent, and then, if the virus changes the kind of target, then do you kind of have a provision for that kind of a contingency situation using technology?
Yes, exactly. We are, I mean, as of now, I would say technology is not being used as much. It is more a method where you have banks of phages, and from your banks or repository, you try different phages against the strain. And if you can find, you develop them as therapeutics, or they might already be clinical grade. But we are saying the prospect, as he asked, now AI will be used, and some companies are using, like in the US, there was a company called Adaptive Phage Therapeutics, which is now taken by Nexabiome. So they are using the information already available on databases to choose which phage would be a good match. And then, based on the efficacy, you can pick more. So you can pick several of those candidates. So these are not used as much, to my knowledge. So far, we are going by the traditional method of phage susceptibility testing. But these are very important applications of AI. And now we are even making phages by AI. So this will be, this is not being done as much, but I'm sure it will be the norm in maybe in coming few years.
First approach, I think, would be in the diagnostics field for just establishing the right sniper to use. Basically, so using the sniper example, you're establishing who's the right sniper for this bacteria. And then gradually, even in phage design, it would have an impact.
I just wanted to ask about, I'm a first year biotechnology student and my minor is in public policy. So you talked about just policy and how the dialogue around phage therapy is very disoriented. So I wanted to ask your opinion on what kind of policies you think would maybe help regulate phage therapy in the future, or AMR. Your opinion on just policies, what do you think should be in place?
So policy in terms of, can phages be used? Because someone has to approve their uses. As Pranav mentioned, as of now, they are being used for last resort cases. Someone has got terribly sick, and then, you're allowed to use as a investigational drug, as we call it. But once the phages are like, it's right now, someone has to take lead. So whether phage researchers should take lead or CDSCO should lead, that's the question. But the policy will be, one, I believe, what should be the standard operating protocols to develop phages as clinical grade phages, because they are safe for administration. Then this debate of, should we go for personalized phages, or should there be GMP? And if GMP is considered important, then who will bear the cost, what will be the cost, and will the cost itself be a deterrent? So these things need to be understood, and most important thing is, like, we are talking phages in the context of antibiotic resistance as a adjunct or replacement to antibiotics, but we have to understand how radically they're different from antibiotics, which are static chemical entity. So before you develop any policy, one has to factor in the nature of phage as a therapeutic. It's biological, it's living, if I may say, or about living. So it responds to infection. So anything which responds is not static. And those things need to be considered while framing policies. And once that happens, it will make life of researchers, all of those who are working, developing banks for these long, they can put to use, and there'll be a kind of direction. What properties should phages have to qualify them as therapeutics? So that one who uses, they are safe and also efficacious.
Can we take the last question there? And I think both Urmi and Pranav will be happy to chat with you after this session. Very sorry, and also sorry to this side of the audience.
Yeah, I'm facing them and I'm not able to take
constantly here. But I hope you still enjoyed this discussion. But yeah, last question.
Hi, can you hear me? Okay. So my question is, you mentioned that intracellular infections are very difficult. Intracellular infections are very difficult to treat currently, right? What do you think is the scope for research in India to genetically engineer the phages to tackle intracellular infections?
I think that's a good question again. we genetically engineer phages can little push back because there's already skepticism about natural viruses. Now, if we engineer them, we have still some time to be there, but we can have, the formulations and encapsulation of phages which enable them to be targeted those intracellular pathogens and so that the cells can internalize. So developing methods so that phages, the cells which are harboring those intracellular pathogens, can they internalize phages when you administer? So more work is needed in that direction. And once that happens and the payload is delivered, then phages know how to do their job. But can they find their way to the pathogen sitting somewhere inside? In mycobacteria, it is macrophage. Inside, there's a granuloma, and then there's an acidic environment. So, they have created a fortress around themselves. So phages have to cross so many channels to get to their pathogen. They say that we will do our job, we will kill them, but take us there. And so taking them there is the thing, which is delivery. And if that is improvised, and I think a lot of drug delivery mechanisms are being developed. Now, can we use them or can we customize for phages? And people have developed already several formulations for this. So we had recently, and not recently, in last 3-4 years, there have been phage therapy for non-tubercular mycobacteria. And there were 20 case studies, and they found mixed results. But that was also telling that, yes, we can go ahead. And now my lab and several others are developing phage cocktails for TB, which is like the most ambitious thing to do. But I feel more about finding specific phages and then delivering them to the target than engineering them for delivery purpose. We may engineer for something else, but for delivery, perhaps, maybe not for now.
Just to add, as of now, like ethics committees, hospital ethics committees, having faced quite a few of them in these Q&A sessions, literally the first question is, are these natural phages, are these genetically GMO phages, genetically modified? So if the answer is latter, then they don't even want to proceed with the discussion any further. So right now, I think there's a lot of resistance to using, yeah.
Thank you so much. Thank you so much, Urmi and Pranav. And I should also thank the audience for such excellent questions, and I know there are many more, so please feel free to get hold of them after this discussion. If phages have piqued your interest, there are 2 very good books that you can pick up. One is The Living Medicine by Lena Zeldovich, and the other one is Good Virus that I mentioned by Tom Ireland. So thank you all for being here, and as we say at Superheroes Against Superbugs, knowledge is power. Knowledge is able to help us take on the challenges that we face today. So stay curious, stay informed, and hope to see you at the next edition, at the next conversation. Thank you.